Hi. This question is related to approval timelines, the review cycles. So the communication during cycle 1 is excellent. We love getting the IRs, the DRLs, so we can get the agency the information they need, to continue the review. But then a CRL comes and then it's silence until the end of the second review cycle, and then a CRL shows up. And sometimes it's related and sometimes it's totally different questions. And I'm wondering would it be possible to get some additional communication in that second or subsequent review cycles?
So what I've heard in terms of the question is enhanced communication during the second, third, the subsequent cycles during review, similar to what we're doing in the first cycle review. I think an aspect, I think that it's great understanding the fact that those additional resources of the staff, they're looking for things at the DRL stage, looking toward those timelines and information. I think we always strive to provide as much, especially through the IRs, as much as possible, and that. I think understanding that, and then as we come to the GDF for, especially having those as priorities, saying that we want to resource and get that additional, I think would be very, very helpful on everybody's sort of part. But understanding is that, yes, we always want to try to provide as much information, but understanding that with the resources we have, we're trying to use this appropriately. I don't know, Diana, did you have, because you're more in the review division and things like that, if you had additional sort of thoughts on that.
Yeah, I think you covered it. It's more of a, we try to communicate as much as we can with IRs, but there's no good of a commitment for DRL in the second and further cycles. So feedback for that going into GDF for negotiations and potential, need for increased resources would be helpful, I think.
Thank you.
All right, our next in-house question.
My question is surrounding discriminating dissolution and exactly what that means to FDA. Do you expect it to fail in the QC media for every parameter or just some parameters? Or can it be out of trend for some of them? And that's very hard to do for an IR product when you just have a single point. So is FDA considering changing like the USP and guidance to say we can have more than one time point on an IR product? And then some of these things you can't get in a controlled correspondence. It seems like FDA is changing their thinking on CBAs and all of that, and all you have is your YouTube videos. And so are you going to change your guidances to actually help direct us on some of these questions?
When you say the one time point, you mean for spec?
Yes.
I review for waiver purpose mainly, but when you show for review purpose, we look at not only one time point, we see multiple time point. We understand for IR drug products, sometimes it's hard to differentiate, but if you show that, we try like different formulation and different manufacturing attribute, sometimes it's not able to differentiate. If you can submit the supporting document and data, then it will be helpful. But for ER drug product, we generally it is nice to easier to show differentiate ability of method.
And I would add, this is, we're bioequivalents, at least the two of us here, but this is, biopharmaceutics would have more to say on this, but I think frequently they do hear justification for very fast releasing, immediate release products, why it can't be discriminatory. And so that's something that would be dealt with by biopharm.
I mean, and I understand that, I'm not trying to put you guys on the spot, but when you say discriminating, do you just mean differences? Because we show differences and then we keep getting the same questions, and it feels like they're saying it must fail the QC method. And that's very hard to do, so just to take that back, that, we just need clarification on what you mean by discriminating in a guidance that actually says discriminating dissolution, we mean it must fail, or something like that, because you can show differences in that, but that doesn't seem to be adequate for them. And like you said, for an IR product, it's very hard to do that when you only have one time point. So just something that we need clarification on. Thank you.
All right, thank you for that. And if we have any more in-house questions, I encourage you to step up to the mic at this time. We'll go ahead and switch to an online question, but by all means, please hang up by the microphone and we'll get to you at the next round.
Okay, this question is directed towards Dr. Vivian. How likely is it that low exposure data in a BE study is due to lack of compliance when BE studies are closely monitored with checks of the oral cavity to assure that the study subjects have ingested the drug product? In most cases, low exposure data cannot be verified or confirmed as to the reason for it. Example, subject non-compliance versus formulation failure.
Yeah, it can be hard to say definitively, and that's why the limitations exist for really the AUC that you're seeing has to be below a certain amount. And it's, again, this is typically for IR products, so this is things that would not be expected to really have formulation issues where they're not releasing. So that was taken into account. This was all internationally negotiated as part of the ICH M13A guidance. So it really was identified as an issue that is happening, and we are seeing that in some cases there is non-compliance, and we've seen that in the past too with periods where there's just no concentration observed at all. It could be due to maybe there's a mouth tag, but then somebody didn't report vomiting a little bit later. So there could be different things that realistically do happen. And so this is a risk-based approach to kind of identify which of those, based on the product type and based on the data that you're seeing, are likely to actually be due to non-compliance.
Thank you.
Excellent, thank you. We'll take our next in-house question.
I need advice, because in PSG it is given, follow this dissolution as given in the FDA dissolution database. And once we follow that, then the question comes, this is not a discriminating dissolution method. So you generate your own discriminating dissolution method. If that is not acceptable to agency, then why there is a recommendation in the FDA database?
I think that's really good feedback. Maybe we can look at the language of the PSGs if that's an issue where it's not as simple. But I think the PSGs do talk about developing methods if needed.
And maybe also a little bit more clarity. Are you getting these sort of deficiencies in terms of for product quality as to routine QC, or are these a part of like the waivers and things like that?
We have got in both the cases, means, yeah.
Okay, and I do know in terms of, especially from a QC perspective, the dissolution website and database is sort of what they consider as a starting point for you guys to be able to use. It's not necessarily the definitive. And I think what the aspect there is, as you develop your product, we also want to see that you've done due diligence in terms of your development of your dissolution, that being a starting point, providing your rationale and additional information there. But I think as Diane also mentioned, that feedback is also very good for us to basically read back. But I think in terms of for your immediate sort of recommendations is always make sure when you're putting your ANDA together is we start with this, we did this root test, and then providing additional justification and rationale so why you felt this is fit for purpose as a starting point for that product.
Yeah, thank you so much. This is helpful. Means if we can put that language in the PSG, I think that will help all of us. Thank you.
Thank you for that. We'll take our next in-house question. And by all means, if you have any, please feel free to step up to the microphone at any time. We have time here.
This question is for Kim. So you mentioned we need all strengths dissolution data to get the waiver. So coming to if you have one product has marketing for different flavors. So in that case, so do you recommend 3 lots under the each strength dissolution required?
Usually, yes.
So 3 lots.
3 lots.
Multimedia as well as QC media.
QC, yes. But yeah.
In QC, definitely. Multimedia, we don't, I think, case by case if there's an issue or something, but we don't always recommend. We have to give, yeah, Xiaojun says no. One should be okay for multimedia, but
One
QC
lot.
3, yeah.
Okay, thank you.
Of course, and we'll go ahead and switch over to our next online question, please.
Thank you. This is for Dr. Kozak. For an expedited ANDA specifically for drug on shortage lists being an IV infusion, what are the expected timelines?
So the question is with regard to a IV drug shortage and expected timelines for the review.
Correct.
I don't have that information off the top of my head. I do know that we have general sort of guidances that talk about the expedited and what those components would be. So I wouldn't necessarily be able to address that question. I don't know if one of my other sort of colleagues would know that sort of process or information. Now, we can get back to you on that, maybe a reply back
Okay.
is through another method, but I don't have that information off my hand. I don't want to provide advice. I don't.
Okay, thank you. I think Tony here has an answer for us.
In the GDF 3 commitments, regardless of what type of drug product it is, a priority application, if it's a brand new application, will receive an 8-month goal date. If that's the question, that's the way I read it.
Yeah. Thank you.
Excellent, thank you for that response. We'll go ahead and take our next in-house question here.
Hi, this is for Dr. Diana. So regarding removal of the data, like low exposure. So if we want to correspond to you before submission of NDA, can we do a control correspondence on it?
Maybe for general questions, if you're asking for pre-acceptance of the data, that may not be appropriate in a control correspondence. You'd have to look at all of the evidence. So I would submit a justification in your ANDA if you already conducted a study. Or if you're wondering like what to put in your protocol or something like that, then sure, if it's before the study, you can do a control correspondence.
So post CRL, can we have a meeting on this one otherwise, if we get a CRL on that one?
You can definitely send a control correspondence post-CRL. It depends on if you're asking for clarification. We have the post-CR clarification meetings available. There are certain restrictions for post-CR scientific meetings because there's a longer review time of these are usually more complicated scientific questions. So it depends on what your question is specifically, if it would allow for that.
Thank you.
Of course. And again, just a reminder, anybody in-house with questions, you're here, the mic's here, panelists are here, feel free to step up. We'll go ahead and take a question via online.
Thank you. Addressed to Dr. Kim. For the case study of metoprolol succinate, the score line is functional, but wouldn't that affect the ER properties of the tablets?
For that drug product, as I presented in the presentation, the film coating was just for easier swallowing, not functional coating. So when the even is split it, the ER function is not compromised for that RLD.
Thank you.
Thank you.
Of course. Any takers in-house? Don't think you're safe just because you're behind that other pillar. Anybody? Okay, we'll go ahead and take our next online question then, and we'll keep going until someone steps up.
Okay. For Dr. Fan, can you please briefly explain why the lowest dose should be used in the comparability BE study?
Okay, so the question is if I can provide more explanation about the lowest dose that should be used in the comparative clinical endpoint study. So we treat it case by case. So I would highly recommend that you check that from our product-specific guidance for the recommendation for each drug products. And if you couldn't find product-specific guidance online, for the drug products, that I would highly recommend that you communicate with us in the pre and communication routes, such as the request a meeting or submit a control correspondence with your questions. Thank you.
Thank you very much. Let's see. They're trickling in, so have patience with me, please. Let's. Okay, this question I believe is for Dr. Vivian. Is ICH M13 presentation applicable for the sublingual route as well?
If you're seeing this specific issue, it could be, I think generally this route is not covered by ICH M15A, is that right? Yeah, but if you have this specific issue, like I mentioned, it could be applicable based on scientific principles for other dosage forms also. So you could, come in with some kind of justification why it could apply to your product.
Thank you. Next question, I think, not quite sure who's the most appropriate person to respond, but here's the question. Will ANDA application result in RTR if applicant used its own discriminatory dissolution method and submit data instead of using OGD's dissolution method recommended in PSG?
I can probably take the first stab at that one. As we mentioned, PSGs are our current recommendations and thinking. You can always use an alternative technique and method, but when submitting in the ANDA, provide justification and or rationale as to why you're deviating from our current thinking and recommendations. So long as you provide that additional sort of statement there of, here's the rationale, we're using an alternative method and this sort of aspect, it will, that usually is what would trigger an RTR if not added. You need to add that additional sort of statement in there that you are using the alternative technique. Then it gets into the review stage. It'll be found at that sort of component if it's acceptable or not. But that's one of the key sort of components. Whenever deviating from a product-specific guidance, please provide that additional information that you are recognizing and that additional rationale. So that way we understand the fact that you are doing a deviation from our current thinking and recommendations.
Thank you. Next question goes to Dr. Vivian. In addition to low exposure data that may indicate subject non-compliance, it's somewhat similar to one we had before, but a little bit different. So in addition to low exposure data that may indicate subject non-compliance, can data be removed from statistical analysis because of real-time documentation of protocol deviations?
Yes, that previous practice is still applicable. If there are protocol deviations that were pre-specified that were not met, that your protocol says to exclude because of that, that can still happen. Of course, I think the most clear-cut thing to do is if you observe a protocol deviation like that in real time, you just don't analyze the data, and then there's really no questions.
Thank you. Question for Dr. Kozak, please. If you can you please elaborate a bit more on the nature of facility deficiencies? Also, are these inspection related?
So I'm going to actually defer to our OP colleagues. I believe we've got to talk a little bit later today about that, but I wouldn't have the additional sort of details on what those major manufacturing facilities are at this time.
Okay, thank you. The next question for Dr. Kim, please. Could you share your insights on whether the common deficiencies presented today are specific to ER products or if they are in fact general deficiencies across all oral dosage forms?
I think there are some deficiency related to ER specific, like alcohol dose dumping or multi-pH media dissolution testing, or score, something like that, that is more like ER specific. But I think most of deficiency is common to IR, which I think is relates to the submission quality, which means can avoid it. Thank you.
Thank you.
And just a quick call for any in-house questions. We still have time. The mics are still here.
Okay, next online question for Dr. Fan, please. Does FDA have a public list of drug products for which a comparative clinical endpoint study is required? We realize that for some drug, oh no, I'm sorry.
Okay.
Yeah,
So-
I think,
Okay, so the question is if FDA has a public list for the comparative clinical endpoint bioequivalence study is required. So the answer is no, we don't have such a public list. However, we do have the product-specific guidance website that's clearly listing whether the clinical endpoint bioequivalence study is recommended or not. So I would highly recommend that you check our website and to see if the clinical endpoint bioequivalence study is needed for the drug products that you are interested to develop. Thank you.
Thank you. Okay, this question is for Dr. Kozak. What is the current first cycle approval rate off the top of your head for generics? And can you provide distinction between complex and non-complex products?
Yeah, actually on my graph, I provided at least some first cycle approval rates for both fiscal year 2018, 2022, and I think we're still fairly close. So 2022 and 2023 would be the most recent data. First cycle approval rates are approximately about 20%, and then second cycle about 20%. With respect to complex versus non-complex, we did look at, and off the top of my head, I do have that for complex products, second cycle approval was at least 25%. So we still are seeing as complex products typically require more than 1 to 2 cycles for approval due to the complexity. However, we're still seeing a good 25% within at least second cycle. Obviously, what we can still do is look at more recent sort of things to get that sort of newer thing, especially as we're putting GDF3 implementation. You know, you get more meetings and things like that. We're hoping and aiming to strive for more first and second cycle approvals.
Thank you. Moving on. Next question for Dr. Kim, please. Could you provide some details on control correspondence submission to clarify the acceptability of inactive ingredient level?
Sure. Yeah, you can clarify the acceptability of inactive ingredient through the CC, control correspondence. Briefly, you can submit 3 questions per each control correspondence, like 3 evaluation, and each inactive ingredient and proposed level, each proposed level will count as 1 question. So for example, like 3 inactive ingredients, 1 level, or 1 inactive ingredient with 3 level will count 3 questions. And some drug product indicate for the pediatric, then will have multiple dosing level, then dose range, then each dose range will count as 1 question. So something like this. So, but I think had the control correspondence session was yesterday. If didn't attend, then I think there is a pre-recording, so you can check, and also control correspondence guidance is available, so more detailed information can be found there. Thank you.
Thank you. And for that inquirer, for everyone, the YouTube link from yesterday is on a one-stop website, so you can easily refer to that while it's still fresh. It looks like this is our last online question, and this goes to Dr. Fan. We realize that for some drugs, it is hard to demonstrate the superiority for the treatment arms to the placebo in the comparative clinical endpoint BE studies. Can you give us some recommendations on how best to handle this situation?
Okay, thank you for the question. The question is, we noticed that there are some challenges for demonstrating superiority to the placebo in a comparative clinical endpoint bioequivalence study. So we understand that it could be a challenge, and we know sometimes it's very hard to demonstrate the superiority to the placebo for some of the drug products, and it could be due to different reasons, and we handle it case by case right now. So I would highly recommend that you communicate with us as soon as you realize the challenge and issue, and could request a meeting, like submit a meeting request or control correspondence, so we can give you specific recommendations. And also in the end of submission, we highly recommend that you provide all the information that you think will be helpful for us to review, such as any supporting documents that you think will be valuable, or any literature that information that you think that will be helpful for your application. Thank you.
Thank you. We have one more that's straight in. Hope it didn't lose its way. This is for Dr. Fan. This is regarding transdermal products. Even if we perform PK endpoint study alone, we will measure the adhesion score during PK endpoint study as well. Can we omit the adhesion study?
Can you repeat the question?
Yeah, I hope, I'm reading, so I hope it makes sense. This is regarding transdermal products. Even if we, I think that word is perform, yeah. Even if we perform PK endpoint study alone, we will measure the adhesion score during PK endpoint study as well. Can we omit the adhesion study?
If the purpose is just for the PK, if you want to omit the adhesion study, so the results is going to be just based on the PK. But for the transdermal drug products approval, adhesion is our recommended study that you would have to do to get your generic drug products approval. So if you don't want to do that in the combined PK and adhesion study, you will have to do another adhesion alone study to evaluate the adhesion performance for your drug products. Thank you.
Thank you much. That is all that we have online. Sorry.
All right, any final last questions here in-house?
I've actually got a real quick plug
Of
too. One
course.
of my colleagues reminded me that for all those people that are interested actually in the IVBT sort of studies, we're having a Center for Research on Complex Generics, April 29th and 23rd is our next workshop on that. So if you are working on those sort of products, that's another good resource that we've got going. Sorry.
No, awesome. Thank you, Dr. Kozak. And just as another resource, we also do have a selection of guidance documents with QR codes out front. So if anything you heard this morning or anything referred now and you want to have a quick resource, check out that table. You can scan those QR codes with your phone and just take a look at one of the ones. I went through and pulled the ones that were mentioned in these presentations today. If I missed any, my apologies, but hopefully those will be helpful. I think now we are good to go ahead and first take a moment to say thank you to our panelists and presenters today.